All right, we can start and maybe people will check. All right.
Okay.
Hello, everybody. Thank you for joining in on the Marine Institute Graduate Society Seminar Series. We meet every week on Wednesdays at noon. Hopefully we can start having these things in person again soon, but I'll let you know. This is being recorded and will be uploaded to our YouTube channel. And that's where you can find almost all of our seminars over the past couple of years. Now, we'd like to provide a land acknowledgement for Newfoundland and Labrador. We respectfully acknowledge the territory in which we gather as the ancestral homelands of the Beothuk, and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We would also like to recognize the Inuit of Nunatsiavut and Nunatuckavut, and the Innu of Nitassinan and their ancestors as the original peoples of Labrador. We strive for respectful relationships with all the peoples of this province, and as we search for collective healing and true reconciliation and honor this beautiful land together. Now, please join me in welcoming DFO research scientist, Dr. Cortney Watt-Wheeler. Dr. Watt graduated with her Ph.D. in biology from the University of Manitoba, working with narwhals. In 2015, she was a postdoctoral fellow, apparently not for very long, at UBC, studying humpback, fin, and killer whale foraging behavior, which is super cool still. Today, Dr. Watt primarily works with marine mammals in the Arctic, studying their ecology, physiology, behavior, and population dynamics. And this is just me reading through her ResearchGate, profile. It was really neat. She's quite literally doing the research that so many marine biology students dream of. And that's not to gloss over the challenges or anything. But with that, I'll hand it over to you, Dr. Watt.
Hi, everyone. Yeah, as Colin mentioned, my name's Cortney Watt, and I'm a research scientist with Fisheries and Oceans Canada here in Winnipeg on Treaty 1 territory. And I just want to say thanks to the graduate students for inviting me to give this talk. And Colin was very generous in that he said I could talk about anything, which was great, but also hard because I'm not sure what is most interesting to you guys. But basically, I decided to give you an overview of kind of the work that I do in the stock assessment group at Fisheries and Oceans Canada. And I know that a lot of you work on stock assessment of different fisheries. And so I think there's maybe some connections there. And I'm happy if you want to interrupt during the talk with any questions, or of course, I'm happy to field questions at the end too. But if you have questions along the way, feel free to interrupt. So this is kind of a general overview, and I do highlight a few like science findings that we've had, but basically I'm going to give you an overview of how we do stock assessments on Arctic marine mammals. And so, okay. So there are Arctic marine mammals across, I work in Eastern Canada around the Nunavut area, and there's an annual subsistence harvest of marine mammals, including whales and seals. And this subsistence harvest is not only important for economics, food security, but also for cultural reasons. And so because of this annual harvest, we need to ensure that the populations that are being harvested are sustainable and that there's a sustainable management framework. And so in the Arctic, we manage animals as discrete units that we call stocks. And these discrete units and stocks are sometimes defined based on genetics, but we also use other indices like movement, summering aggregations, and starting to look at chemical signatures to also define stocks. So why do we study stocks? Well, it helps us to understand population structure and natural history of the species, but stocks are officially management units, and it also helps us ensure that there's not local extirpation of marine mammals around communities where hunters rely on them. And so the first and probably primary goal of stock assessment of marine mammals is we need to know how many there are. And so we do regular abundance estimates. And when I say regular, I know a lot of the fisheries science, they're able to do surveys annually. And we don't do annual surveys on every one of our stocks, partly because these are long-lived animals, and so they have much longer generation times. But we do strive to update abundance estimates on our stocks every 5 years. Sometimes we fail on that, and it's a little bit longer between stock assessments, but that's the goal. And so how do we do abundance estimates on marine mammals? We typically have flown in twin otter airplanes where we have observers sitting both in the front and back of the plane, basically with their head outside of a bubble window looking down and counting whales. And then we can only cover sort of a small transect under and to either side of the airplane, and then we extrapolate those numbers to and the densities of animals to areas that we aren't able to cover and see in the twin otter, but we're able to cover quite large areas with this method. At the same time, we have a camera running in the belly of the twin otter that is continuously taking photos. And so we've also based abundance estimates off of photographic surveys where we count whales in photos after the survey. And that image in the bottom is just showing how we communicate with the pilots to develop our surveys using ForeFlight. And so the pilots follow strict survey lines for us to be able to then extrapolate and get density and abundance estimates for large areas. There are other ways to get abundance estimates, which we are using more and more, but not currently used in any of the marine mammal stock assessments in the Arctic, but they are supplementing information on abundance, and this includes genetic mark recapture. This typically works for smaller populations. So we started using this for bowhead whales in the Canadian Arctic that visit similar areas every summer. So we go out and basically use a crossbow or an air gun to shoot a biopsy dart at a bowhead whale. And this image shows a whale with a dart on it, and then the dart bounces off and floats in the water, and then you can collect it after. And what you get is shown in this other image is a little sample of skin, and sometimes if you have a direct shot, you'll also get some blubber on that sample. And we can use those samples for a lot of different things that I'm going to talk about throughout the talk. But for genetic mark recapture, we can identify this individual genetically, and then we can see if we recapture that individual in subsequent years. And then you can use a formula that looks like this to estimate the population abundance. So that's genetic mark recapture, which is somewhat invasive because you're having to shoot a dart and collect a little skin sample on these whales, but we can also use photographic mark recapture. And so this is a photo of a whale that isn't in the Arctic that has a nice dorsal fin, and you can see that this dorsal fin is quite marked up. It's got notches on it, and so we can use that to identify individuals and then match them up across years. But this is more difficult in the Arctic because we have animals lack a dorsal fin. And so we have to use other indicators, but there are other indicators to look at marks to be able to get a photographic mark recapture. And I think I have some images of what we've been doing recently is using drone technology. So these are 3 different drones that we've used to take photographs and video of mostly beluga whales and bowhead whales in the Canadian Arctic. And we've been collecting photos for a number of years this way on a small population of beluga, which I'll talk about in a bit, but they're called the Cumberland Sound beluga population, and they're estimated at between 1,000 and 1,500 individuals. So relatively small, and we've been going out every summer and taking images of them with drones to be able to develop this photographic mark recapture abundance estimate. And this just shows how we fly the drone from a small zodiac, and you can catch it on the water, and it's quite minimally invasive. Sometimes the whales we've found that they will respond and turn and look at the drone, but typically we fly them 20 meters or higher because we found that there's no adverse effects or no reactions of the whales when the drones are flown at that height. So this just gives you an example of an animal that has quite a distinct scar mark that we've been able to identify 3 years in a row. From 2017 to 2019, came back to the same summering area every year. So these are two other methods you can get at abundance estimation. And then a new method that I've started using is to use satellite imagery to monitor marine mammals in the Arctic. And so we started collecting imagery in 2018 in a summering area that is known to have a high aggregation of beluga. And from there, we just wanted to ground truth if we could actually identify beluga and narwhal from satellite images. And we published this paper in PLOS One last year that showed that we could identify them. And then this program has kind of expanded quite a bit because with COVID, unfortunately, we were unable to go up and do a twin otter survey on some of the narwhal and beluga stocks that we were hoping to. And so instead, we tasked the WorldView 3 satellite to take very high resolution imagery of these areas. And so from this, we were able to identify whales and count them. And so I'm not sure how well it shows on your screen, but this is a satellite image with a number of beluga whales in it in an estuary in the high Arctic. And I will come back to this also. And so how else do we monitor and distinguish stocks? So the first thing is we need an abundance estimate of the stock, but how else can we ensure that the stocks are discrete units, and how do we determine what is a stock? And so we use in part movement ecology. So we satellite tag marine mammals across the Canadian Arctic, and in my job I mostly work on narwhal and beluga. So we've been tagging, DFO has been tagging these whales with these satellite link transmitters since the 1980s. And I think worldwide we've tagged over 100 narwhal from West Greenland and Eastern Canada with these satellite tags. So this gives us pretty good information on not just their location, but also dive information and environmental information. So these tags keep track of the temperature of the water that the whale's in, the salinity, and then gives you information on their dive profiles and then of course their location when they're at the surface of the water. And so although we have a lot of whales tagged, these tags, you can see these tags in particular are backpack tags, and they're the whale is captured in a net, and then these backpack tags are secured on with those pins, and it's kind of like a piercing that goes through their dorsal ridge and holds these satellite transmitters on. And they last quite a bit longer than other tagging methods, but we've only had, I think, 2 tags that have lasted a full year cycle. So this gives us information about site fidelity within the summering season, and then information on their migration to their wintering grounds, but it hasn't provided us with information on an annual basis as to if the whales continually return back to the same summering grounds. We've only had 2 tags that have lasted that long. But this has provided us significant information on narwhal and beluga movement. And now my lab, we're trying to move to less invasive methods for monitoring whales. And so this is another method to tag potentially narwhal and beluga. We haven't been successful with narwhal using this method yet, but we were successful with beluga. And so this method, you use a crossbow or an air rifle to shoot a tag onto the whale. So you don't have to capture or handle the whale, and the tags go into the blubber and through the skin and into the blubber, and then you can see they kind of splay the 2 anchor points, splay in the animal's tissue and remain attached. The downside of this method is that we don't get nearly as long tracks and information because the tags work their way out a lot sooner than they do with the pin attachment. But we had success this summer deploying one of these tags on beluga, and I have a little video which I hope will play in this mode. So you can see the boats just parked and the engine's off while they kind of wait for a beluga to surface. And this is a community-based program that's running in Pang, Nunavut. And so you can see they just shot the tank. So this is considered quite a bit less invasive than the capture and handle tagging that DFO has historically done. So this is what the data looks like. And you're like, well, what's going on here? Why is there beluga on land? So this is the issue with the tags. They have to make contact with a number of satellites to get really accurate points. And so this I'm just showing you because this hasn't been cleaned, this data. So these points are basically, they've only made an uplink with a single satellite. And so they're able to say like, okay, the tag's somewhere around here on Baffin Island, but we don't really know where. So for analyses purposes, we would remove these points because they don't have high enough accuracy to be able to really tell us anything about the position of the whale. But these positions are much higher accuracy, and they would also be accompanied with dive data, because if you get multiple uplinks with multiple satellites, then there's more time and more satellites to transmit that data. Whereas these very low quality positional data points wouldn't have had dive data associated with them. So yeah, this is just real time. This is from November 2021. So we haven't analyzed this data fully yet, but that was just from this past summer. And then we also use genetic signatures to monitor stocks. And I'm not a geneticist, but I am keenly interested in discriminating stocks based on genetics. And it's been very difficult with narwhal, because narwhal were believed to have gone through a population bottleneck historically. And so that resulted in them having very low genetic diversity. So even though there's quite a few what we think of as discrete units based on their movements and based on the localities of the hunting regions and the whales coming back to those areas year after year, there's not a lot of discrimination among those stocks. And so this just shows a PCA of the genetics of some different narwhal stocks and populations in the Canadian Arctic and in Greenland. And so you can see that this was able to separate out. There are 3 populations of narwhal worldwide that are genetically distinct. So there's the East Greenland population that's pulling out there as yellow, the Northern Hudson Bay population. So this is in Northern Hudson Bay, Canada, around the communities of Nowit and communities along the Hudson Strait. And then there's this Baffin Bay, what we call the Baffin Bay population. And this population on this side, you can see what we call different stocks, that those are Somerset, Jones, East Baffin, Inglefield, Bredning in Greenland. We call these different stocks, but genetically they're all very similar and we can't discriminate among them. And so that's why we need to use other techniques for separating these into stocks. However, this publication actually is getting quite old. It's from 2011, and recent analyses in East Greenland narwhal have shown that with whole genome sequencing, they're actually able to discriminate stocks better. And so there's a current study underway doing whole genome sequencing of some of these Baffin Bay stocks. And we're hoping that there may be a little bit more genetic discrimination with the whole genome sequencing. And then for stock assessment models, it's nice to know the age structure and the sex structure of the harvest, because then you can incorporate that into your population dynamics model. And age and sex structured models are probably what you see a lot in fisheries, in fishery stock assessments. Unfortunately, we don't get a ton of the harvest of marine mammals sampled, but we do have a community-based marine mammal sampling program that runs across 21 communities in Nunavut, but it's voluntary, and hunters basically pick up a sample kit, which is just a kit with everything labeled and different bags, and it asks for different tissues from the animals. And you can imagine that these are large animals and it's difficult to get all the tissues. So sometimes we get kits back that are only, half filled. Sometimes we do get really full complete kits, and then some hunters don't participate in the program or don't have kits on them. And so we only get a small proportion of the harvest sampled. But when we do get an aging structure like a tooth or an eye, we try to assess the age of that animal based on these structures. And so you can get the age of an animal from their tooth. It's somewhat akin to the tree rings that you would see in a tree trunk, where you can count a ring every year. And so in teeth, if you subsection them, you can look at annual growth layer groups, we call them, but basically a line in the tooth indicates a season or a year in that life. And so you can count them up and estimate the age of the animal. This is pretty labor intensive because you have to cut the tooth in half, you have to subsection the tooth, and then you have to polish it and really try to bring out those lines. And then someone has to read this tooth under a microscope. And so we are doing that work historically at DFO, but we don't have anyone who ages whales. And so we've been sending them to a lab for aging analysis. You can also envision that we don't necessarily always get teeth because they are harder to cut the jaw out of the animal after the harvest. And so sometimes we also don't get the teeth. The eye is easier to collect, and we've started using aspartic acid racemization of the eye lens to look at age of the animals. And so basically there's these enantiomers in the eye, and they change from D to L shape. I don't know if you guys remember your organic chem, but they change from the D to L isomer over time. And so if you get an estimate of what the ratio of those two is in a fetus, then you can compare the ratio of older whales and get an age estimate. Of course, there's an error associated with that, but the eye is a little bit easier to collect. And then very recently, we've started looking into epigenetic clock age estimation. And this is something that Dr. Steve Horvath has been doing in the US, using basically, can we age people based on their DNA and their telomere length? And there's caveats with this also, because, you know, if you're, say, a smoker or a drinker, you might shorten your telomeres faster than if you're someone who only eats fruits and vegetables and works out all the time. So your lifestyle might also impact how quickly your telomeres shorten. And so we're starting to investigate this with beluga whales. First, to say, can we get an estimated age from the epigenetics, but also, is there differences in how quickly whales are aging depending on where they are and what kind of anthropogenic stressors they might be experiencing. So you can imagine whales that are constantly around ships, like maybe in the St. Lawrence estuary, they're maybe dealing with a little bit higher stress from that, and that might prematurely age them compared to other populations that aren't undergoing that stress. And then, of course, all the populations are dealing with impacts of climate change. And so that might also have an impact on how quickly it looks like their DNA is aging them. So we're trying right now to combine all three of these methods, counting lines in teeth, aspartic acid racemization of the eyelids, and the epigenetic aging from skin tissue, which, if that works, could be the sort of easiest method for estimating age of the whales. And then I've also looked at chemical signatures to monitor stocks. And so I've used various chemical signatures, including stable isotopes. And stable isotopes, you've probably heard of, but a lot of people use nitrogen and carbon to evaluate what animals eat. And so nitrogen can tell you something about the trophic level of an animal, because you are what you eat, and with some enrichment. So whatever you eat plus an enrichment factor is the estimated nitrogen that you can have. And that's a very simplistic version of it. But you can use models to estimate an animal's diet if you know the nitrogen and carbon signatures of their prey and the nitrogen and carbon signatures of the predator, and you know something about what kind of enrichment happens as they lay down these signatures in their tissues. And so I've used stable isotopes, including nitrogen and carbon, and carbon, this is just showing, so nitrogen gives you a really good idea of the trophic level, and carbon gives you a really good idea of where the carbon source of the prey that they're eating is coming from. And so you can see just in this plot, it shows that planktonic carbon has a much lower signature than benthic carbon. So you can get an estimate of whether they're feeding more in a benthic food web versus a pelagic food web. Stable isotopes also vary with location and geography. And so this shows how they vary, nitrogen and carbon vary with latitude. So this is useful for monitoring stocks in theory, because if their signatures are going to vary depending on where they are, then we can say, okay, well, if these animals are in the same area every year and the signatures vary from other areas, we should be able to say, like, yes, these whales stay in this area. It's complicated by this because these marine mammals migrate, some of them quite large distances, and so then they're picking up stable isotope signatures potentially from other areas also. And so there's definitely some confounding factors with this, but it is an interesting method that we're starting to use. And we've had success with discriminating stocks, narwhal stocks with stable isotopes, even better than we've had with genetics. So it is a promising avenue, but there are a lot of caveats with it. And then we've been using fatty acids to look at diet. So fatty acids, similar to stable isotopes, you are what you eat. So you lay down a similar fatty acid signature as your main prey. And so if you're eating different prey in different areas, you might also have different fatty acid signatures. And the fatty acids are measured from the blubber samples that we get from marine mammal sampling programs, but also that you can get from those biopsies that I mentioned you can take. And then trace elements basically occur in very small quantities, and they come from your diet or your environment, and they also vary spatially. So similar to stable isotopes, we can use, if a specific fjord or inlet has a specific trace element signature, then we can try to tie an animal back to this area. And so we're doing this currently. I've done it in soft tissue, in skin tissue, but we're doing it currently in tusk and tooth material to look at trace elements to see. The nice thing about a tooth is that I mentioned they lay down an annual signal. And so you can measure the trace elements in each of those little growth layer groups to see if the trace element signature varies across years, or does it remain the same. So if an animal goes back to the same area every single year, then their trace element signature every year at that time should be similar. And so we're trying to look at that particularly for narwhals. And so I've already really talked about, but I'm going to get more specific into some of the work I've been doing on narwhal and beluga in the Canadian Arctic. And those are the two species I work on mostly. So for narwhal, there's 3 populations in the world. This shows their distribution in dark gray, light gray, and black. So 2 of those are in Canada. The Baffin Bay population has about 140,000 narwhal, and their winter distribution is shown with the hashed lines, and then their summer distribution with hashed. And then light gray is the Northern Hudson Bay population, and that population, well, it was estimated at about 12,000, I think, when I put this talk together, but we have had a recent abundance estimate that suggests it's more closer to 18,000, 19,000. And so the Northern Hudson Bay narwhal, the last estimate was from 2011, and we estimated that there were 12,500 individuals, but we did complete a new survey in August 2018. This is the last kind of survey that I've done on narwhal or beluga that I could talk to you about. So it seems a little bit old, but we were unable to do surveys in 2020 and 2021. And I didn't run a survey in 2019. So this is the last survey we did in Northern Hudson Bay of this population. And you can see these were the different areas we flew with a twin otter airplane with 4 observers, each perched out a window, a bubble window in the twin otter. And those lines on the map just show the transect lines that the airplane flew to develop this abundance estimate. And we also had the camera running in the camera hatch, and you can see this is what the images look like. So we saw both narwhal and beluga on this survey, but beluga are less common there, and the goal of the survey was to survey narwhal, but we ask our surveyors to note any other marine mammals that they see, and so we also were able to see some beluga in this survey. And then, so that's the Northern Hudson Bay population and a survey that we ran there. And then there's also the Baffin Bay population. And this population, I mentioned, quite a bit larger and divided into, so the whole Northern Hudson Bay population kind of summers in that area. And so by surveying that small area, we're able to get an estimate of the population abundance. The population of narwhal in Baffin Bay is quite a bit larger, and they all spend winter in a similar area in Baffin Bay and Davis Strait, but then in the summer they migrate to all these fjords and inlets in northern Canada and West Greenland. And so this complicates things because they're spread out. You can see the Somerset Island, that's SI in the blue, that's their estimated summer distribution. And basically that's a massive distribution, and we can't survey all that area, and we're basing that whole area based on traditional knowledge from hunters that say, hey, we've seen narwhal here, and we say, okay, they must be part of the Somerset Island stock. But I'll be honest that we know relatively little about the way all the narwhal that inhabit that area, and are they segregated within that massive area? We're not sure, still to be determined. Then some of the other stocks are a little bit smaller distribution, but still quite large. And then of course you have to time these surveys so that you get them in the summering area at the time when they migrate in there. So they're sort of kept out of some of these areas because of sea ice. And what ends up happening is they overwinter in Baffin Bay, and then they kind of follow the ice edge further north as it moves. And then as the ice melts and dissipates, they move into these summering areas. And so there was a massive survey of all these stocks, which was quite the undertaking because it was 4 different planes. And even then the survey took over a month. So sometimes an area was surveyed 2 weeks before another area. And in theory, the whales can move between these areas. So there is, we hope that they don't move much in the summering aggregation period, but it's always a possibility that there is some movement. But these are the last estimates we have for narwhal stocks in the Canadian high Arctic. And then added up, we get about 140,000 animals in living in Canadian waters. Oh, sorry, I skipped that one. Okay, so then I also mentioned we have like a long history of tagging narwhal, mostly in the Baffin Bay population, but we have tagged some whales in Northern Hudson Bay also. And this comes from tagging studies that were done in 2006 and 2007. And we have used those tagging studies to identify the wintering and summering grounds of the Northern Hudson Bay narwhal. But in this particular paper, I looked at their diving behavior and to identify maybe important foraging areas for the populations. And so essentially, I argued that if they're diving right to the bottom, narwhal are known to to gain a lot of their energy reserves from Greenland halibut. And so Greenland halibut are in quite deep water. And so I argue that if whales are diving almost to the bottom, I'm going to assume that's a fairly energetically expensive dive, and they're likely doing it to forage. And so because of that, I looked at the home range for all their dives, and you can see it's kind of hard to see on the migration, but it does look like they might be foraging a little bit even on the migration. And then you can see this is their summering area on the top and their wintering area on the bottom in blue. And then I just looked at the areas where they're making those really deep dives and said, okay, within the areas where they're doing all their diving, when are they going right to the bottom? Because that's when I'm going to hone in on for when I think that they're actually foraging. And so then this shows a reduced area of the home range for just what I would call their critical foraging habitat. So this is useful to identify both their home range and where they spend their time in their migration, but also potential areas that could be focused on for conservation targets for conserving critical foraging habitat. And then we've had a lot of tags that we've looked at from various stocks of narwhal in the Baffin Bay population, and over 100 whales have been tagged, and I've only been involved in a small subset of those, but this shows some whales that we tagged in Arctic Bay and near Pond Inlet in 2010, 11, and 12. And so you can see their summer is in the top right. They're quite condensed in those summering areas. And then in the fall, they move out, start moving out and making their migration to the wintering areas. Then you can see in the winter, they're mostly in the Baffin Bay region. And then by spring, we don't have very many tags left. You can see, I think there's only 2 there. And you can see that they start to head back to kind of their summering grounds. And so this is where we've got a lot of our information about the stocks and how to separate them is based on the tagging data. We actually have a large management model that considers movement of the whales, and it's complicated because it's a management model that crosses international boundaries, because whales don't care about international boundaries. So this is a model that's been developed and promoted, and it's not currently in use, but we have published on it, and we're hoping that both Canada and Greenland will move this way. It's been developed by both Canadian and Greenlandic scientists working on narwhal, and it basically considers all the tags that we've ever had and all the movement of whales through different hunting regions, and it estimates a probability of how many whales from each stock might be available to hunters in different regions. And it's complicated, but essentially you can see all the numbers, like the ratio or the fractions show how many tags out of the total number of whales that have been tagged from that stock might be available to hunters in other areas. And so we're trying to use this as an integrated international management model that would consider movement of whales and takes in 2 different countries. And we're not quite there yet, but we're moving towards it. And then I mentioned counting growth layer groups in teeth, and for narwhals particularly, they don't have any teeth. They only have the sort of iconic tusk that you think of when you hear about narwhals. And those tusks are expensive and sometimes hard to get a hold of because hunters like to keep them. They use them for carving and for craft purposes. And they're quite expensive and valuable. So they're hard for us to get a hold of. But along with the tusk that erupts through the left side of the jaw, there's often an embedded tusk that remains embedded inside the jaw on the right side. And females have 2 of these embedded tusks. So females usually don't end up having an erupted tusk, so they have 2 of these embedded tusks. So we did ask hunters to collect these embedded tusks for us if they were willing to as part of the sampling program. And so we did get quite a few embedded tusks back. And we use these embedded tusks just like a tooth. We cut them open to age the whales because we really don't have much information on age of narwhal, because historically we used the teeth to count, and it's difficult to get teeth from narwhals. And then we started using aspartic acid racemization of the eye lens, but we needed to ground truth that with something. So to use the aspartic acid racemization, you have to get first information on the true age of the whale and compare their AAR values to that to create a curve that you can then estimate age for. And so we did this back in 2018 and published a paper on it, developing a curve for estimating narwhal age using aspartic acid racemization for animals for which we also had embedded tusks where we could age them with GLGs. And so I mentioned stable isotopes have been working to separate populations and stocks. And so this plot just shows the stable isotopes of nitrogen and carbon in Baffin Bay narwhal versus northern Hudson Bay narwhal. And you can see that they're quite separated, and geographically they're separated, but the stable isotopes were even more distinguished than we would expect by geography alone. So this suggested not only are stable isotopes telling us something about the geographic segregation of these animals, but maybe about their diet as well. And so we did some isotope mixing models and found out that it seems like northern Hudson Bay males and females are foraging more on a benthic diet that compared to pelagic, whereas the Baffin Bay whales are foraging approximately equally in both a pelagic and benthic food web. So this told us a little bit more about the diet of those whales, and especially about what food webs they're relying on. But then we tried to look specifically at different prey, and this arose specifically because the fishermen in different areas were wondering, well, how much shrimp or how much halibut are narwhal consuming in these areas? And I won't go into this too much because there's a lot of caveats with the isotope mixing models and how distinguishable the different prey are. But we tried to do some modeling to get an estimate. And we found that Baffin Bay and northern Hudson Bay have some squid and capelin in their diet. Northern Hudson Bay has a greater proportion of halibut in their diet than the other populations, whereas Baffin Bay is relying on those pelagic Arctic cod and polar cod species. And then we looked at if diet changes seasonally. So the interesting thing about stable isotopes is if you look at different tissues, they turn over at different rates. And so you can compare, for instance, stable isotope values of muscle tissues, which represent a lot longer time frame than skin, and see if you can pick up any seasonal differences in diet. And so we did find that there were significant differences in the diet of Baffin Bay and Baffin Bay males and females in what we thought represented the last 6 months of diet as opposed to just the last 2 months of diet. And this might be reflective of their migration and also foraging on different prey depending on where they are. And we saw less distinguishing difference between muscle and skin in northern Hudson Bay narwhal, and they have a smaller migration area and also seem to rely on more similar prey in the different seasons. And then you can look at differences over time also, and this can be complicated because there's an impact of anthropogenic carbon entering the food webs, which has changed carbon values over time, but you can try to correct for that and look at if there's been any temporal changes in diet over time. And we did see a little bit of a change in northern Hudson Bay in nitrogen, but no change in the Baffin Bay whales. And yeah, I'm not going to go into too much more detail about that because I think I'm starting to push my time, but we're also looking at stable isotopes and trace elements now in tusks. And so we've acquired embedded tusks and also some full-size tusks. And then basically we're drilling powder, drilling the tooth into a powder along each growth layer group, and then running the analyses on those individual growth layer groups. And this has provided us information on diet and individual variability in diet. And we had a paper just accepted this morning in Marine Mammal Science that looks at individual variability. But this paper on weaning age also came out last year. And so basically, if you're consuming your mother's tissues, it's you're drinking her milk. It's like you're consuming her tissues and you're at a higher trophic level. And so you can pinpoint when a narwhal is weaned from its mom based on its stable isotope signature. And so we make assumptions in our stock assessment models about how often narwhal reproduce, and we try to rely on traditional knowledge for that because we don't have a really good handle on the narwhal's reproductive cycle. But based on this, we used weaning age as one method to look at how often animals wean, or how long do animals nurse their young, and if we can assume that they're nursing their young and not reproducing for the first few years of that nursing, then we can try to estimate a reproductive cycle. And we actually found there was a lot more variability in nursing duration than we expected, with some narwhal nursing into their 5th year. So most of the whales nursed for 2 years, but we saw variation in that 2 years. Yeah, and then we're also looking at dietary variation among individuals. So you get a number of measurements of diet signal throughout an animal's life, and you can see how variable they are across individuals. And this is interesting because narwhals were considered quite specialized in terms of their diet. And we're actually finding out that there might be a little bit more variability and adaptability possibility for these animals as climate change changes their food webs. We are finding that there are sort of little niches that different animals take advantage of. And so there's a bit more variation in their diet than was originally thought. And then I mentioned we've used trace elements to separate stocks, and there's some overlap, but you can see each of these ellipses represents what we've defined as a stock based on movement and hunting localities that see these whales year after year. And so there is variation in the trace element signatures among some of the stocks, and although there is overlap in some, this actually offered more separation than the genetic signatures have. So this was optimistic in using trace elements, and this was in skin and soft tissues. And so the graph on the left shows the summering samples from the summer, where we define them as summering stocks. And so we expect differences, and we see more differences there compared to the other plot, which shows, sorry, you can't see my mouse probably. The other plot, which shows the spring samples that were harvested in the spring, and there's a little bit more overlap, and you might expect that there would be because then whales are on their migration and there's some mixing. And so that's kind of an overview of what we're doing on narwhal, what I've been doing recently with narwhal. And now the other stock that I work a lot on is Cumberland Sound Beluga. Cumberland Sound Beluga spend their summer in the northern part of Cumberland Sound in an area called Clearwater Fiord. And then they spend winter just at the mouth of Cumberland Sound. So they have a very small distribution. And this is primarily based on satellite tagging data. They have a small distribution compared to the Baffin Bay narwhal. And so we did a survey estimate of these guys in 2014 using an aerial twin otter survey. And the 2017 survey, I'm sorry, I didn't update this. The 2014 survey was an estimate of 1150. In the 20s, we did another survey in 2017, and the analysis is done, and we've estimated around 1,300 individuals. But the confidence intervals on those abundance estimates overlapped significantly. So yeah, this population is estimated between about 1,000 and 1,500 individuals. Whales are known to congregate in Clearwater Fiord, and so this is a good opportunity to be able to photograph a high proportion of them. And we've been using full coverage photographic surveys where we just fly the twin otter across the entire fjord area and photograph the whole thing, and then have someone count all those images. It's also a really great area to collect satellite imagery. And so we've been collecting satellite imagery in this area since 2018 every year to look at variation in the distribution and abundance within this fjord. And this is also the area where we've been developing a photographic identification catalog using drone photography. And the drone photography has some benefits because you get an overhead image of both sides of the whale at the same time. So you don't have to develop a catalog for the left side of the animal and a catalog for the right side of the animal. And so this is another animal with a distinguishing mark that we recaptured 2 years in a row. And we were able to also have a community, this is a community-led program, and we were able to have more photographs collected in this past summer in 2021. And so we're analyzing those photos right now to compare to the current photo ID catalog that we've developed. And then we're also starting a boat-based photo ID catalog, which is taking images from the side. And this one is a little bit longer to develop because you do need both that left side and right side catalog. And then beluga haven't been tagged in this area since 2006 to 2008 when 7 beluga were tagged, and they provided information on the summer migration and winter distribution of the whales, and they also provided information on the dive behavior. And so these are areas where whales were diving more than 50 meters, and so then presuming to be foraging. And we've also looked at the stable isotopes we've also looked at stable isotopes in this population to compare diet, and found that the diet may vary across the different age groups of beluga. And so there might be some ontogenetic shifts in diet. And then we've looked at stable isotopes over time, which have also changed and suggested that the whales may be consuming a greater proportion of capelin compared to Arctic cod. And this is kind of a new phenomenon, I guess. I shouldn't say new, because I think there's always been capelin in this area, but capelin have never developed and had consistent spawning grounds there. And now in recent years, in the last 10 to 15 years, community members are seeing capelin spawning in Cumberland Sound every year. And it does seem, when we're looking at both the fatty acids and the stable isotopes from these whales that are spending their summers in this area, they are able to take advantage of those capelin spawning individuals, and they do seem to be consuming a greater proportion of capelin and less Arctic cod over time. Then we've looked at their diving behavior, which just shows essentially that they're really making those deep dives in the winter, and that might be a really important time for them to gain a lot of their energy reserves is on the wintering grounds. And that capelin are found in this shallow area, and they may be foraging more on capelin in the summer. So yeah, this was just a little graphic to show the hypotheses about halibut's the main diet in the autumn and winter. They're consuming a little bit of cod all year round, and then mainly that capelin, then they're taking advantage of those spawning grounds in the summer. And then finally, I've been working on the eastern high Arctic Baffin Bay beluga population a little bit, and this population overwinters in the North Water and Baffin Bay area, and then spends summer around Somerset Island. And there hasn't been a lot of work on this beluga population. It's not harvested heavily in Canada, but it is harvested in Greenland. But we haven't tagged whales from this population since 1996, and this just shows a little graphic of the tracks of where the whales moved. And we used these tracks as evidence of some of the estuaries that we thought would be inhabited by beluga in the summer. And there hasn't been an abundance estimate for this population since 1996. And so when we were grounded in 2020 due to COVID, we took advantage of the WorldView 3 satellite, and we tasked it to take images of all these little orange areas. We tasked all these images of all these estuaries to try to get a new abundance estimate of belugas from the high Arctic. And there are issues with the satellite, the cloud cover problems, and also the timing of the images, and whether they can image a whole estuary in a single shot or not depends on the location and the strip. But overall, I'll say that it worked fairly well, and we were able to get density estimates of beluga in these different estuaries from the images, and we use crowdsourcers to read the imagery. And so we're still currently doing QA, QC on all the crowdsourcing. But basically the images were put out to a crowd, and they could go and tag whales in the image. And then this was done a minimum of 3 times for each tile. So the images were all divided into little squares that could pop up easily on the crowdsourcer's computer screen, and then they could click to post a location of a whale and then move to a new tile. And so we're doing a lot of QA QC to see how happy we are with how well the crowd kind of did. But the preliminary estimates were quite similar to what was found in the 1996 aerial survey estimate of about 21,000 beluga whales. And so we're not sure if that's a good thing yet, because we were hoping this population would have increased in size, but we only covered the estuaries and none of the open water areas with the satellite. And so I would say that this should be a minimum estimate, but I think it shows some promise. And I think that's everything I was going to talk to you guys about today. And I'm sorry, I tried to rush at the end there to leave some time for questions.
That's okay, as long as you have time to sit around and answer.
But
But
yeah, I have some
yeah,
time.
thank you so much for that. That was great. Does anybody have any questions? You can go ahead and raise your hand, type in the chat, or just go ahead and speak up. I was gonna ask, um, what's the incentive for the hunters to like? Is there a different incentive for each program? Is there only one program? I kind of wasn't sure. You kept talking about the hunting program, but I wasn't sure if there's separate ones for like narwhals versus the other.
Yeah, so we run it as one big marine mammal sampling program, and they basically, so we'll take samples from seals, walrus, whales, any subsistent harvested species. And the incentive is that we compensate them for their time. So and it varies a little bit. So a seal kit is worth $100, and a whale kit is worth anywhere between $100 and $200, depending on if they also collect the reproductive tract. So there is a little bit of compensation for sharing their food, but it's more to compensate for the time that it takes them to fill in the sampling kit. And then we do all the so I'm a scientist for DFO, but all the science advice that we provide for sustainable management, that isn't necessarily what's accepted and what's decided upon. The management of marine mammals in Arctic Canada is considered a co-management framework. And so we're all kind of contributing pieces to the pie of this co-management framework. And so there's a lot of collaboration with the hunters and trappers. And so with the Hunters and Trappers organizations and the Nunavut Wildlife Management Board and Fisheries and Oceans Canada. And so I think there's also incentive there to participate in that sort of co-management framework. And we use a lot of the samples that they provide to provide information into the stock assessments.
So is there, I mean, you mentioned those other organizations. Are there many universities up there doing the same work? Do they provide you with any of that data, or do you use any data from their results?
Yeah, so any university can request. So Nunavut does have the Arctic College, and sometimes there are students in the Arctic College interested, and we take them on as like co-op students or field guides and contractors to help with the field work. In terms of other universities, I would say there are like universities that are doing research in the Arctic, and we encourage them to make sure that that research is collaborative and consulted on with the communities. And if they particularly are looking for samples, because we've had this program running for a long time, we have a huge archive of samples, and the samples we DFO, you can apply to basically come and get the samples and run whatever analyses you want on them. But we ask for an application because we want to make sure that the community that provided the samples is consulted about the work that's going to be done, and then also not just consulted about the work that's going to be done, but then told the results of that, because we don't like the communities to hear in, for instance, in the news, something's going on with samples they've provided and no one's kind of ever told them that they're working on that. So yeah, the samples are really an archive of, to me, the Canadian marine mammal history. And so they're an important archive, but they are accessible for researchers. And we've had lots of different university collaborations where they've relied on the samples from the program.
Sure. I was kind of wondering about the satellite tags. You had shown a picture of them, and I just kept thinking of how much those must cost. Because you said they don't last. You've only had two last a full year. Um, so when you put them on, you must just hold a little honorary, RIP tag. Your life expectancy. You have less than a year to survive. There
Yeah,
goes thousands of dollars, I'm guessing.
yeah, yeah. Yeah, they vary in price, but I think the most recent ones that we're shooting on the whales are about 7,000 Canadian each. So it is
And
not.
then that doesn't count the boat ride. If you're catching them, take. I'm assuming you have to have like a fisherman or somebody who you charter, and then everybody to handle a big whale or some type of whale. But
Yeah,
yeah, it must really add up. So the funding on that must be tough to
yeah.
come by.
The funding it's piecing together a lot of different pots of money, but I will say that narwhal are kind of a high priority for fisheries and oceans. So we have been successful in accessing funds to be able to run the programs, but for sure they're really expensive programs to run. And I know one year we went all the way to Grise Fiord, which is the highest community and the most northern community in Canada. And we all were there for 3 weeks with tags and we never saw narwhal. So then there's also that, where that was a very expensive program and we got like literally not a single data point
Oh my
about
gosh.
it. So yeah, it is a risky endeavor, but the yeah, the shooting the tags on, it requires a much smaller team of people, and this past year it was entirely community based with researchers in the community. So in terms of that, I guess it was more fiscally responsible, but still the tags themselves alone are yeah, very expensive.
It's really interesting because in the fisheries world, when we do stock assessments, it's based on a really small really small sample, right? So I'm guessing that you guys have a lot more ability to get a larger like proportion in your sample for your stock assessments. It just feels like that when you're talking about, leaning over and counting and doing the surveys and doing transects and stuff like that. I was just assuming that you're counting a larger proportion than we're usually getting like 0.0001%, and we're basing the entire community off of that. So
Right. Yeah, what can I say about that? We are missing, so the complicated thing with whales is we're only seeing them when they're at the surface of the water and they spend a lot of time below the water, and we're not like trawling or fishing. So we try to use those satellite tags. I guess I didn't mention this, but we try to use those satellite tags to adjust our survey estimates to consider how much time whales spend at the surface versus how much time they spend at depth. And there's a lot of error associated with that. So we're making an assumption that we're, in some cases missing like 3/4 of the whales because they're under the water, depending on the area we're in and the tags. And sometimes we're using tag data from 10 years earlier for a survey that's happening now. And it's like, well, maybe the behavior of the whales has changed. Maybe the behavior of the whale changes every day and we're using an average. And like we kind of hope that it averages out and that we're somewhere in the ballpark. But I've heard that, we shouldn't really, I call them abundance estimates, but they are really indexes of abundance.
I see. All right. Well, I think, yeah, nobody else had any questions. I think I'm guessing everybody really understands stable isotopes in here, so nobody had any questions.
Yeah, and I'm
Yeah,
sorry I kind of breezed through that. I tried to cover a lot in a short, but so if anyone has questions, you can feel free to email me after too. My email's just courtney.watt@dfo-mpo.gc.ca, and I'm sure Colin can provide it if you have
yes,
questions.
absolutely. All right. Well, thank you so much for coming in and talking to us, Dr. Watt. Before I end it, I do want to introduce next week's seminar. Dr. Anna Braswell from the University of Florida will be joining us via WebEx. Dr. Braswell will be discussing coastal ecology and how coastal development is tackled when considering ecosystem needs. And once again, thank you, Dr. Watt, and everyone for joining us today.
Thanks everyone for having me. Take care.
You too.